Okay, let's get started. Hi everyone, my name is Yuju Lee. I'm a second year computer science student at the University of Toronto. Today I'm presenting my work on segmentation error-aware probabilistic clustering for highly multiplex imaging reveals densely packed tissue dynamics. ISC is a type of multiplex imaging data. It could measure 40 or more protein expressions in parallel. The workflow involves antibodies coupled with mellow tags to stain tissues and then bind to proteins. And these tags will go through the cytometers to assemble images. While INC provides spatial information and enables fixing tissues of region of interest, its image has relatively low resolution, captures small spatial areas, and requires segmenting cells. The figure on the left is a stack of protein images, and the right is the combined protein images into one. Therefore, a cell segmentation must apply on these images, and it is a process of identifying individual cells considering all given protein images. After segmentation, mean expression in each cell is used as single-cell data. The figure below denotes segmentation mask, where each red outline is a cell. However, doublets are common segmentation errors. A doublet is composed of multiple cells but identified as a single cell. This confounds the biological interpretation of cell populations. Here I will show some possible issues. The first figure shows multiple nuclei in a mass cell inside the red outline. The scatter plot illustrates B and T cells co-expressions. And the figure on the right comes from a densely packed tissue, which may affect neighboring cell expression levels. These examples suggest doublet exist. As a result, it motivates us to develop a model to identify cell populations in INC while accounting for segmentation errors. The model we developed is called Starling. We use protein expression and cell segmentation results to construct a single-cell dataset. The model considers single and doublet likelihoods and their assignments. Finally, the model provides doublet probabilities and singlet clusters. We further use three metrics to evaluate our model: the accuracy from the validation data set of the simulated data, the possibility score, which I will discuss later, and the morphological clustering score by comparing the predictive singlet and doublet sets using t-test. Starling stands for segmentation-aware clustering. The key idea is that it modifies standard mixture model by introducing additional indicator variables for cell n. Dn denotes is cell n a doublet. Zn means if Dn is a singlet, which cluster does it belong to? Gamma signifies if Dn is a doublet, which two clusters does it assign to? Here I will introduce Starling more formally. There are three priors. D assumed to be a Bernoulli distribution. Z and gamma assumed to be categorical distributions because they are assigning cells to clusters. Y denotes protein expression, and S means cell size. They both assume to be normal distribution or T distributions. In the doublet case, we model the average of expression and sum of cell size. Note that the sum of two independent Gaussian is a Gaussian. The model loss is between likelihood from the data and the simulated data. We train the model using mini batching and other optimization. For this work, we generated densely packed human tonsil samples. There are 3 samples and comprise of 16 regions of interest as part of the larger oncologic resection of oral carcinomas. Each sample consists of 24 markers, which is shown on the left. and about 200,000 cells retrieved after applying Mesmer, a cell segmentation algorithm. In the remaining of the talk, we will show the model results based on 50,000 subsamples. We compute the cluster possibility score by considering two cases. The first case is when protein should not be co-expressed, shown in the figure on the left. The other case is when protein 2 only expressed when protein 1 is. For the tonsil data, we selected 25 protein pairs shown in the table on the right. For each pair, we compare or compute the 25 and 75% quantiles of each protein expressions, and the frequency of centroids fall into the expected regions depending on the cases. The final score is the sum of all 25 pairs. Here we compare the cluster possibility score shown in y-axis of 4 clustering methods showing x-axis relative to various lambdas, the regularization term, and cofactor, which is our sync the input data. In our experiment, we run 5 random starts of each model variant. We compute the score at initialization, showing the blue box plots, and after training Starling, showing the orange box plots. Overall, score seems to increase after training Starling. Here we show the result of detecting synthetic doublets from the simulated data. The data was randomly selected from the observed data. There are equal number of singlets and doublets in the set. For the purpose of evaluation, we generate training and validation sets. And the figure on the right is the result from the validation set. We output F1 score shown in orange box plot, area under ROC, and precision recall curves shown in blue and green box plots. Overall score seems to stabilize at lambda equals to 5 with F1 score close to 90%. Currently, we are investigating how Starling centroids compares to other clustering methods, as there is identifiability issues among clustering results. Starling uses GAN initialization, which is shown in the bottom left plot. The Starling centroid plot on the right is based on lambda 1 and cofactor 5. Starling seems to suggest clusters 16 to 20 as doublet clusters because no cells are assigning to them. The preliminary results suggest Starling identified cleaner cell types and possible cell types that are not revealed in other methods. As for future directions, we plan to extend the model to beyond doublets, include spatial information in the model, and develop user-friendly Python package for everyone to use. Finally, I would like to acknowledge everyone in my lab, especially my supervisor, Kieran Campbell, our collaborators, and our funding partners. Thank you.